Humans have been on the search for immortality for thousands of years. Our knowledge about aging and longevity has changed dramatically over the past century. One of the greatest contributors to the current study of aging, longevity, and lifespan was Dr. Leonard Hayflick, whose revolutionary paper, co-authored in 1961, flipped the field of aging research on its head when he discovered that cells were not immortal, as scientists had believed, but that they had a replication limit, now named after him, the Hayflick limit. This discovery of cell senescence became the foundation of modern aging research. Dr. Hayflick's cell lines went on to be the basis of safe vaccinations that have saved millions of lives. It was a huge honor this past year to be able to have a conversation with Dr. Hayflick, looking back on his career, his contributions, and the momentous discoveries that he had the dedication to pursue. This episode is dedicated to the memory of Dr. Hayflick, who recently passed away at the age of 96. I hope you enjoy the conversation, and thank you for listening.
What advice would you give to wrap us up here for the young people who are interested in biomedical research, who could only dream of being able to look back on a career with the kind of impact that you have had?
Question everything.
Dr. Hayflick, you have had a legendary career in the study of aging and cellular biology. Can you start us off and tell us a little bit about what it's like to look back on a career that has had this much of a global impact?
Well, I'm not sure I can agree with your premise, but when I look back at my career, excuse me, just a minute. I am proud of the accomplishments that I have made in respect to the discovery of cellular senescence and the discovery that only cancer cells are immortal and that normal human and animal cells are not immortal, and my discovery of the cause of human primary atypical pneumonia, better known as walking pneumonia, when I discovered the cause of it was a new mycoplasma that I named Mycoplasma pneumoniae. And the effect that my work on aging has had in reorienting the belief once held that aging, the cause of aging was in extracellular phenomena like radiation, stress, things like that. But my work has oriented the notion to direct attention to intracellular events as the cause of aging, and in particular to state that the cause of aging is not a biological phenomena but a physical phenomena, and that it is caused by the second law of thermodynamics, where entropy increases and the stability of biomolecules cannot be maintained forever because they are the subject of the second law. I don't know how much more detail you want me to go into. I'm assuming that we will do that in our discussion.
It's hard to participate in the modern biomedical research community and imagine what it would have been like to have had the perception that people did have before 1962. The idea that aging was purely an external process happening, and this notion that you mentioned that had been previously held in the previous half of the 20th century, that if a cell could simply be in a cell culture medium with the proper provisions, it could simply and would simply be immortal. How different would that drive medical research to assume that that was the case, as opposed to the now universally accepted recognition after your work that, in fact, even with perfect cell cultures, cells are mortal and limited in their replication capacity?
Well, the belief prior to my work was completely wrong. Normal cells have a limited capacity for replication. They do not replicate indefinitely, despite what alleged evidence might have occurred. There was absolutely no evidence that normal cells have a finite capacity to replicate, including the work of Alexis Carrel, which I and several others disproved.
Now, this was the French biologist who had forwarded the claim that using cultured cells derived from embryonic chickens, that he was able to prove that cultured cells were in fact immortal. Was he the primary actor promoting the incorrect proposition that cells were immortal?
Yes, I think that's fair to say, and I and several others proved that that was nonsense, and that his work was a failure because the repetitive feeding of his allegedly immortal culture of sheep heart fibroblasts was reseeded every time the culture was fed with no fibroblasts that existed, and the embryo extract that was incorrectly prepared and allowed for the existence of new young cell fibroblasts being delivered to the allegedly immortal culture every time it was fed.
Now, that is fascinating. So you identified an experimental error that was propagating this indefinitely in his cell culture medium. Did this get accepted by the research community? Because I know you were a young researcher. You didn't have what now is extensive worldwide fame in this field. What was it like demonstrating and advancing research that showed that the primary assumption of cellular immortality was wrong when you took it to the research community for publication?
Well, when I took it for publication, the publication was rejected by the Journal of Experimental Medicine, where the paper was first sent. It was rejected by a man who won the Nobel Prize two years later. And that was, of course, a disappointment, but we then sent the paper directly to Experimental Cell Research, and they published this almost immediately without change. The people who rejected it at the Rockefeller Institute, where the Journal of Experimental Medicine was published, were part of the old guard who simply believed the notion that when cells are put in culture with perfect under perfect culture conditions, that they would replicate indefinitely, which we proved was absolutely false.
I can imagine that the primary medical interest in reducing the mortality of living patients, of living organisms, would have a very different focus under the idea that cells were naturally immortal. It would make sense to kind of chase down one after the other external variable, whether radiation or infection. Were there proposals that humans would begin to reach dramatically longer lifespans with simply addressing radiation control and infection control before you were able to demonstrate that aging was in fact an internal process?
Well, I don't recollect in detail whether any of them had that belief, other than a fringe group of people who have always existed in this field ever since it began 3,500 years ago, with the belief that certain things like waters from various sources produce immortality in humans. But that belief has been with human civilization ever since recorded history, about 5,000 years ago. It's nothing new, it still exists. There's a lunatic fringe that still believes it. That's because they have no understanding of the etiology of human aging and all aging. Human aging is no different than the aging of non-biological materials, because aging is the result of the second law, which applies to everything in the universe, including the universe itself. Nothing is immortal. And I challenge anyone to provide evidence to the contrary.
You've made in line with that observation that these stochastic processes disrupt the flow of available energy. You've made some interesting observations about the presence of aging in non-human species that are maintained in the absence of predators, for example, in zoo collections of wild animals. You've also pointed out that aging should be distinctly thought of from the process that actually gives us longevity. And I think that you've done a really interesting thing in pointing out the difference, because a lot of times when we're discussing optimizing lifespan, there's a very quick tendency to blend together ideas about the diseases of aging and the lifespan of organisms in a way that doesn't clarify that the diseases that actually cause death in later life don't necessarily resolve aging itself. And you once wrote that even though influenza used to take many lives of the elderly, once it was resolved, it didn't teach us about the actual mechanisms of aging. Why is it so important to point out that those are two different things?
Because it has shown, even as of today, that there is no organization in the United States, and probably the rest of the world, but at least in the United States, because I'm aware of what's going on better here than what's going on out of this country. And that is that no institution in the United States with the word aging in its title, including the National Institute on Aging, of which I was a founding member, has its research directed toward the etiology of aging. All institutes an organization as with aging in their title, is doing research on age-associated diseases, the resolution of which will not provide us with any information about the fundamental etiology of aging. So this is a huge scandal. I challenge anyone to show me an institution in this country who has major research is devoted to determine the cause of aging. And I challenge anyone to provide the name of an organization that's doing that. All organizations are doing the same thing. They're studying age-associated diseases. None of the revolution of no age-associated disease, including the reading courses, will provide us with information on the basic fundamental biology of the cause of aging. It just does not exist in this country, despite all of the noise in which people are claiming that their particular organization is studying aging. They're not studying aging. They're studying age-associated disease. Which is a completely different universe. It has nothing to do with aging. It is expressed as animals age because of the second law, but it's not the etiology of aging.
So if tomorrow we could wake up and we had a biomedical solution that prevented a single person from dying from cancer, or a single person from dying from heart disease or Alzheimer's, we would still not be informed about the actual drivers of aging itself. Is that fair?
Yes, of course. If all of today's age-associated diseases were resolved, we could not live longer than with a life expectation greater than about 92. It's impossible. If every cause of disease, age-associated disease, was resolved, including cancer, cardiovascular disease, stroke, even Alzheimer's, which is a trivial cause of a large number of deaths. And then the elimination of all of those leading causes of death would only result in a life expectation of about 92 years, and that's been shown time and time again. People constantly ignore it.
I think that's a fascinating observation, because we see in very wealthy countries in the West, especially in very high income earning countries in Europe that have nearly total access to the most advanced healthcare available, that this indeed tends to still be a limiting lifespan. And we really have a lot of channels in media and medicine and research, and of course, a lot of marketers with a lot of products to sell a very desperate population who's interested in living longer, who's interested in escaping aging. And yet, despite all of this, despite billions of dollars being sunk into it, and despite profits in the billions of dollars for companies that are becoming wealthy on the desperation for a resolution to aging, we still don't have answers to the fundamentals of aging.
Yes, that's because nobody's studying it. Very simple answer.
And do you think that that is something
I interfere with longevity determinants? And although they are to some extent manipulable, aging is not manipulable. It is an absolute certainty. Because you cannot violate a principle of physics. aging is a problem in physics and not biology.
Now, this is an important distinction, because when we talk about the biology of aging, it has been proposed at times that we could perhaps look for a genetic program that might programmatically limit the lifespan of a species. And you're suggesting that this is definitively the wrong approach, that the answer does not lie in biology, does not lie in a genetic program, because that stochastic degradation of cellular function is, as you say, a matter of physics and not biology.
Well, genetics has nothing to do with aging. It's been shown time and time again. I challenge anyone to prove or provide evidence that genetics plays a role in the aging phenomenon. It definitely plays a role in longevity determination, which can be extended by doing all of the things that most of us are aware of in respect to maintaining life. But genetics has nothing to do directly with aging, other than the exposure of the molecules that form the genetic processes, like genes and every other molecule in an animal's body that suffers from the same fate that is provided by the second law. It's a principle of physics. You can't violate it.
Now, should we be looking for, in regard to splitting up the research and the conceptualization of aging into these separate anabolic and catabolic processes, the anabolic process of extending longevity as opposed to the catabolic processes of breaking down cell function over time through aging, do you feel like there is more need for research into extending the functions that enhance longevity? Or do you think it's more important to first uncover the specific pathways, the specific mechanisms by which the damage of aging occurs? Should we prefer one over the other?
Well, first of all, you have to understand that the desire to increase longevity is a phenomenon that is held almost entirely by people who live in developed countries. There are millions, if not billions, of people who live in underdeveloped countries who would not wish to have their lives extended. So with that helpful understanding, there is, first of all, no way to intervene with the aging process, despite the billions of dollars that have been thrown at it by startup companies today and by rich multi-billionaires who have put money into efforts to do so, which is not a new phenomenon. That has been going on again for as long as we have had written living history, where the rich, powerful have been looking for ways to increase their longevity. Every effort to do so has failed and will continue to fail because there is lack of knowledge about the theology of aging, number one, and number two, all of the research being done allegedly on aging and by organizations with that word in their title is not conducting research on the etiology of aging. So I'll state now, I think for the third or fourth time, that you cannot expect any results or new information being obtained if all the institutions in this country, for example, are studying age-associated diseases and not the fundamental underlying etiology of aging that predisposes biological pathways to age-associated diseases. It's just not going to happen. If you don't do research on a subject, how in the world do you expect it to be understood?
Do you think that the profit motive of focusing on resolving either the aesthetic effects of aging or the diseases associated with aging drives the continued focus on the age-associated disease? Or do you think it just continues to be a misunderstanding that equates the two?
I think it's partially both. The people who are raising money on it, trying to promote or sell things or lifestyles that allegedly could increase longevity in it to make money. That's no surprise.
Should we encourage skepticism?
They have not distinguished between the anabolic process of longevity determination and the catabolic process of aging. They are completely different phenomena. And I doubt that they have the intelligence, most of the money makers or people who want to use their wealth to promote the idea. They have no understanding of the distinction between longevity, determination, and aging. It's that simple.
During your career of navigating this very interesting country where funding oftentimes crosses government boundaries, oftentimes crosses private investment boundaries and things of that sort, do you think that there was a time period in the history of the U.S. when we could have diverted to a more scientific understanding of aging in a way that would have avoided some of the lost research years and wasted funding that would have perhaps given us insight into the necessary research on mechanisms of aging earlier?
Well, of course, I would like to believe that we would have made efforts to understand the fundamental biology of aging before today, but that didn't happen. So I think we can do about what did not happen in the past. The National Institute on Aging, of which I mentioned earlier, I was a founding member of that organization and founding chair of its executive committee, etc. Even to this day, the greatest proportion of funding spent by the National Institute on Aging is on Alzheimer's disease. And the resolution of Alzheimer's disease as a cause of death in this country would add approximately 19 days onto our life expectancy, a trivial number yet. The money spent on Alzheimer's disease research in this country is absolutely astounding. That exceeds the expenditure on every other aspect of longevity determination and aging in this country.
That's fascinating. That's fascinating.
Was unfortunately started by Bob Butler, the first director of the Institute, after I had turned down the offer to be its director. Bob, who was a psychiatrist, promoted research on Alzheimer's disease, which was understandable, but he himself publicly announced years after he promoted the expenditure of funds on Alzheimer's disease that the amount of money had gotten out of hand. So much money is being thrown at research on Alzheimer's disease that is being spent on fewer and fewer qualified people, just like what happened during Nixon's war on cancer. The same phenomenon occurred.
Now, would we see a greater benefit? Because we understand now that the diseases of aging and the resolution of diseases of aging will not drive huge lifespan gains. And we understand that
I beg to differ. That is a false premise. That belief still exists, and the evidence for it is enormous. And I've stated again and again, the evidence is that most of the money spent on what is called aging research, which is a really serious problem, and what those two words mean, that the money is being allegedly spent on aging research is being spent on age-associated diseases.
Right. Yes. This misconception drives the funding towards solving and resolving the ages associated with disease, which doesn't clarify the actual mechanisms of aging. So the question becomes, if the funding were to flip, if we could have a dream scenario where we were able to get the funding into the actual understanding of the mechanisms of aging, do you think it would give us some key insights that would actually also perform the resolution of those diseases that everyone is so keen on trying to solve?
Well, that would be my hope. In fact, I pointed that out in one of the papers that I sent to you to read, and that is that because, in my judgment, aging is a phenomenon in physics and not biology, it is an area of funding for the National Science Foundation and not the NIH. And the National Science Foundation, hopefully, if it would, and it has funded several small conferences on the etiology of aging, unlike the NIH. The National Science Foundation has indeed funded at least two, and I was present at both, meetings that were concerned with the etiology of aging. That has never happened at the NIH. And the hope is that once you understand how old cells differ from young cells in a lineage in respect to their molecular landscape, you might hopefully discover why the molecular landscape in an old cell increases. It provides opportunities for disease to be caused. And that would be a most important development if you could understand why old cells in a lineage are more susceptible to disease than our young cells. That would provide opportunities for intervention in the causation of those diseases. It would have no impact on the cause of aging itself, but it could theoretically provide an understanding, or at least a better understanding, on what kind of landscape, molecular landscape, leads to what we call age-associated diseases and not adult-associated diseases or infant-associated diseases. So it's clearly a problem in logic. You have to look at the molecular biology of old cells to determine why they are more vulnerable to the cause of age-associated diseases. It doesn't take a genius to understand why that is so.
Solve the susceptibility problem and we'll figure out why the diseases are happening instead of trying to cure the diseases after the cells have already become susceptible.
Yes. Why is there such a term as age-associated diseases? Clearly, people that will pronounce those words must have a belief that the phenomenon of aging predisposes cells to disease. It's obvious, it's logical, but nobody studies it.
Wow. And this is...
I mean nobody, and I challenge anyone to provide evidence to the contrary.
And that's not going to change until somebody is able to and willing to put the money where the research is pointing.
It's not going to change also for political reasons, and that is when you approach a politician and ask for money to support research on aging, they look at you with grassy eyes, not understand what you're talking about. But when you approach a politician and say you want money to study cancer, which is age-associated, or cardiovascular disease, or Alzheimer's, which is age-associated, then they understand what you're talking about and they'll fund that. But they won't fund the underlying cause that increases the vulnerability to those diseases about which they clearly understand because they themselves or their loved one that suffers from one of those age-associated diseases. But they do not have the biological background to understand that the fundamental common denominator to all age-associated diseases is age.
It's a fascinating, real human problem that we have, that when we can visualize the problem or the suffering from a specific disease that somebody has had who we know and we care about, we're okay with putting big dollars into trying to find a specific solution. But it is really hard to convince people that understanding the process that led to that susceptibility could have an even more profound, massive implication on preventing an entire next generation from becoming susceptible to those same diseases.
Yes, it's because politicians have no knowledge, and I should say they're ignorant of fundamental biology. 99% of them are. And that's taken advantage of by the people who run organizations with the word aging in their title. It's taken advantage of by them because they know that they need to ask for money for specific diseases and not for the underlying etiology of all of them. And that perpetuates the cycle that has been going on for decades.
Wow. Can you talk to us a little bit about the process of discovery for the young minds who might be listening to this, who are interested in having the kind of impact that changes a field? You wrote once that when you noticed the phenomenon of cell cultures beginning to stop replication after a certain number of cycles, you said it wasn't so much a eureka moment as much as it was a, that's funny moment. And how can young scientists have that kind of attention to detail to find the that's funny moment and turn it into something that became a very carefully crafted series of experiments that ultimately changed the minds of an entire field?
Well, it's because I was taught, like everyone was taught at the time I made that discovery, that when cells are put into culture and provided the proper milieu in vitro, which was the terminology used in the letter that rejected my paper when it was sent to Journal of Experimental Medicine, when you make a finding that has in which there is evidence that the prevailing beliefs are not correct. You have to have the courage to go with it and try to find out why what you found seems to defy what was taught to you. And that's exactly what happened when I made this observation. Like everyone that's in the cell culture, I was taught, as I mentioned several times, that when you provide cells with the proper milieu in vitro, they will replicate forever. Well, I didn't know, of course, that I was providing the cells with everything they needed to replicate. It turned out in retrospect that I did. But when I found that one of several cultures had cells in it that stopped dividing, I immediately assumed that it was my fault and having made some error in culture conditions. But then when I found second and third cultures derived from second and third tissues, then it became apparent that this was becoming a common phenomenon and that I should pay attention to it, which I did do. And in fact, I abandoned my original research, which these cells were to have been used in a study on the etiology of cancer. When I observed that the cultures grown from different tissues, from different fetuses, had a limitation on their ability to divide, that directed my attention to that problem exclusively, and I abandoned the research on cancer that I described. So you have to have the courage to go ahead with what you observed and where you have sufficient evidence to believe that it is not something that's artifactual, that it's actually true. And you pursue it, which is what I did. And there's a lot of the paper being that I originally wrote, being rejected for the same reason that I was taught. People who rejected my paper said exactly what I thought I had disproven. They said that if you provide cell in vitro, those are the exact words, that the cells will divide forever. Well, that was not true. That was proven in my paper, and proven in a very elegant way, I must say, by putting into culture equal numbers of male and female normal human cells, which could be distinguished by each other, and then determining that if you mix cultures of young female cells with old male cells, and then carried the unmixed components separately, I found that the culture containing those male and female cells eventually contained only female cells, because the male cells had subdividing as they did in the control vessels. So that had a very significant meaning. It meant that whatever was causing the cessation of replication of the male cells was not a diffusible product that only infected male cells and not female cells. So we don't know anything that has that distinction or makes that distinction. So that was pretty darn close to being absolute evidence that what was occurring was an intracellular phenomena and not an extracellular phenomenon.
And this was a brilliant for any of the young young budding biologists or researchers out there, this was such a brilliant use of the new sort of cell culture and cytogenetic technology that was just coming to bear. The way that you and your research associate were able to use the presence, if I remember right, the presence of bar bodies as a experimentally distinguishable variable to separate the two source cells along the lines of which population had occurred, of which population had replicated more times. And then the way that you sort of crossed the experiment over and did it the other way, it was such a perfectly timed use of what was brand new technology with cytogenetics at that time.
Yes, I think that's a fair statement. Les Paul-Moore's use of the bar body, the female X chromosome that is sustainable during interphase through female cells that allow us to have this essential marker that distinguishes male from female cells. And as I ultimately renamed it, the dirty old male experiment also worked when you did the reverse with a dirty old female experiment. And that proved without a doubt that what was going on was an intracellular phenomenon. Had nothing to do with the birth media or whether the glass was properly washed or whether the moon was on proper phase or not. So the evidence was absolutely clear, but even that evidence resulted in the paper being rejected by the Journal of Experimental Medicine at Rockefeller. And it ultimately was unpersuasive for about 10 years, I would guess, by the biology community who didn't want to believe what I had proven. And that's a very common phenomenon of science, when there is a leap of understanding, of no understanding, that is greater than what is anticipated. And then at that point takes a much longer time when it is true to accept it. It's a phenomenon that's well known in science. When a discovery like that is made, it's at first ridiculed, as mine was ridiculed. And then after people repeat it and it gets published in a few papers, then eyebrows get raised and some people say, well, you know, maybe that's true. And then as more and more people repeat the experiments and get the same results, ultimately it's believed. That took 10 years in my case, which is not unusual. And then after 10 years, as it is now, it is received knowledge. It's no longer even a reference. I find that papers talk about cellular senescence now without any reference to my original work, which again is the way it goes. That's the way.
After, of course, your paper became one of the most cited science papers of all time. I think at last count it was in the thousands and thousands of references, right? It was one of the most cited papers in the entire 1960s and 1970s.
Yes, there are actually two papers, and both papers have been cited close to 20,000 times.
The goal of every young researcher is even to get a few goals in motivated references.
Papers 2 or 3.
Right.
Mine have both together cited 18 some odd thousand times.
That's amazing. And it really, it almost seems that during those first few years, there's this perceived cost to giving up a an understanding of the way something as significant as aging works. There were, of course, the scientists and Nobel laureates like Elie Metchnikoff who had essentially proposed that if we could just reduce infection enough, we could get to the point where we would see really dramatic lifespan extension, because that was just the proposed external forces that were driving aging. And then you showed this, and now cellular senescence is just common received background knowledge. Now, after you went through the very detailed building of these experiments and you became a leader in cell cultures, you went on to have this very large global impact because your cell culture lines did something to the development of vaccines that completely changed and significantly enhanced the safety of vaccine development. Can you tell us what kind of cells, for listeners who aren't familiar with how vaccines used to be developed, what kind of cells were used to develop vaccines before the types of cell lines that you created became dominant in vaccine production?
Well, in fact, there were no cell types that were used for vaccine production before. I showed that my cell strain WI38 and WI26 were used for that purpose. Vaccines were made in embryonic eggs. The antivirus of the vaccine still is, to some extent. So the cell culture essentially was married to virology in the late 1950s and through the 1960s. That marriage resulted in the establishment of several very successful vaccines. One of the first cell cultures that was used was primary or transplanted monkey kidney cells used by Salk and Sabin to grow the viruses that were used in the first polio vaccines. It was then discovered that those monkey kidney cells were contaminated with an endogenous virus with some of which were fatal to humans. And it was about that time that I showed that WI26 and WI38, my normal human diploid fibroblast cell strains, were susceptible to all the then known human viruses, including polio, rubella, measles, mumps, measles, etc. And furthermore, red blood cells were free of contaminating unwanted viruses. And finally, and most importantly, the cells linked to the then used rubella viruses and rabies viruses the important properties that allowed them to be used as vaccine substrates. So people who are gifted by WI38 strain to at the Worcester Institute went around with my gifted cells, which allowed the viruses, the rubella virus and the rabies virus, to grow in a way that produced no side effects to vaccines, and furthermore provided it with properties of attenuation, so that not only did it allow for side effects, but it also gave to the vaccine the necessary necessary ingredient of attenuation. So without my gift of WI-38 to both of those investigators at the Wistar Institute, there would have been no successful rabies vaccine or rubella vaccine that did occur at that time. Unfortunately, those people at the Wistar Institute took out process patents and never told me or included me or Moorhead in those patents. You'll have to ask them why, although they're no longer living, so it would be very difficult, why they kept Moorhead and I ignorant of their efforts to obtain vaccines, which they did do, and they themselves made significant sums of money, and the Wistar Institute to this day makes about $30 million a year on the process patents that they have on the rubella and rabies vaccines. Those patents would not be possible without the use of my WI-38 cells, and that's acknowledged in the first paragraph of both of the patents. Yet the head of the institute and the other people who developed the two vaccines kept Moorhead and I ignorant of the patent applications, and consequently we never received a penny in remuneration, despite the fact that without our gifts there wouldn't have been neither vaccine.
And I think this really speaks to the process of science and the personal investment of years of insight and experimental design, and of course the blood, sweat, and tears and the energy investment that core scientific researchers like yourself and Moorhead put into the processes that have given us the kind of safe vaccines that are almost taken for granted on a daily basis, because it really was a big shift to go from not having cell cultures or from using cell cultures from other species to having safe cultured human cells in order to have these vaccines developed. And it really underlies, even with the personal experience that you guys had of not being included in the patent, that ultimately the scientists who drive the development of these technologies for the betterment of all of us oftentimes not only can't have a profit motive, but in some cases that it is foisted entirely in a different direction. And yet now over 1 billion people have received the benefit of having those safe and available vaccines thanks to that core research that you guys were able to do.
Yes, even to this day, for example, Merck and the company produces a rubella vaccine, either in my cells or a copycat strain, because when I published how to make these cell strains, other people made them, but they have a monopoly on the rubella vaccine made and sold in the Western Hemisphere. Comes from vaccines made in normal human diploid fibroblasts, which is what I originally gave to them as the strain WI-38. As you pointed out, and I think I sent you a copy of the paper, if not, happy to do so. A paper was published a few years ago showing that the vaccines against rubella, rabies, adenovirus, and polio, and hepatitis B. All of the viruses together have benefited closer to a billion people worldwide. Lloyd and I never made a penny from any of those vaccines. The people who did were the people who patented without telling us at the Wistar Institute and by the Wistar Institute itself, even to this day.
What advice would you give to wrap us up here for the young people who are interested in biomedical research, who could only dream of being able to look back on a career with the kind of impact that you have had?
Question everything.
That's powerful. Thank you so much, Dr. Hayflick. Really appreciate you joining us for the conversation today. And of course, thank you for your lifetime of hard work and impact on this field.